Hi, my name is Gabriela Leite. I'm from MAST at Cedars-Sinai Medical Center, and I'm here to present my poster, Higher Levels of Serum Anti-Vinculin Antibodies Correlate with Greater Severity of Gastrointestinal Symptoms and Small Intestinal Bacterial Overgrowth, or SIBO, Microbial Profile based on Quantitative Shotgun Sequencing. Immunizing rats with Campylobacter jejuni cytolithal distending toxin B, or CDTB, results in higher serum antibodies anti-CDTB. Via molecular mimicry with endogenous proteins vinculin, a subset of rats also produce autoantibodies against this molecule. This process is also seen in humans and is linked to small intestinal bacterial overgrowth. Antibodies anti-vinculin quantification is a validated marker of post-infectious IBS in humans, and SIBO is more common in IBS. However, the link between antibodies anti-vinculin and SIBO in humans is still unclear. Thus, we investigate the relationship between antibodies anti-vinculin and SIBO by applying quantitative shotgun sequencing in duodenal microbial samples from human subjects undergoing standard of care upper endoscopy. In this study, we perform microbiome analysis in duodenal aspirates collected in subjects undergoing EDD without colon prep. Serum was also utilized to test for antibodies anti-vinculin, and a cutoff of higher or equal to 1.65 was considered positive. A quantitative shotgun metagenomic sequencing was applied. A total of 614 subjects were analyzed, and 155 of them were positive for antibodies anti-vinculin. Subjects positive for antibodies anti-vinculin had a distinct microbiome profile with significantly lower microbial diversity compared to those that were negative for this antibody, even after correcting for SIBO. The absolute abundance of several disruptors from the genera Escherichia and Klebsiella was higher in subjects positive for antibodies anti-vinculin when compared to those that were negative. The absolute abundance of Escherichia and Klebsiella were highest in double positivity, including SIBO and antibodies anti-vinculin, as compared to those that were negative for both conditions. And this profile correlated with greater severity of symptoms such as diarrhea and urgency. In conclusion, autoimmunity to vinculin is linked to major shifts in the small bowel microbiome, including increased absolute abundance of classic SIBO microbes such as Klebsiella and Escherichia. When subjects were positive for SIBO and antibodies anti-vinculin, these changes were even more pronounced, with greater loss of microbial diversity and higher gram-negative loads and more severe gastrointestinal symptoms. Thank you for listening to my poster, and we have other posters here at DDW 2026. Check them out.